introduction level.
I.G. Amish Srinivas of second M.Pharm of Assurance Department heartily welcome you to all to the day 3 of 3-day national webinar on current strategies in targeted drug delivery in association with Association of Pharmaceutical Teachers of India, APTI, AP State Branch. Now we can have an AV of our college, Sri Vishnu College of Pharmacy. It's my privilege to introduce our today's guest speaker, Dr. Shankar Bhattacharya, Associate Professor, Department of Pharmaceutics, NIMS, School of Pharmacy and Technology Management, Shirpur Campus. Sir has completed his postdoc in Nanomedicine IIT in Banaras Hindu University, Varanasi, 2019. Sir has completed PhD at RK University, Rajkot, 2018. He had completed B.Pharm and M.Pharm in the Department of Pharmaceutics at SRM University, Chennai. Sir has published 43 articles in various national and international journals, and he also published 2 books and 1 book chapter. Sir has done his research project in Gujarat Council of Science and Technology, DST. He received HREA Annual Award in 2020 in the field of research in pharmacy by Human Rights Education Association. He received Global Teaching Excellent Award, that is GITA Award in 2020 in subcategory of 10 Professors of the Year. He received in SC Young Achiever Award by Institute of Scholars, Bangalore on July 1st, 2020. He received Excellence Teaching in Higher Education Award by Sridhar Bahudeshya Vikas Santa on July 2, 2020. His areas of research are nanomedicine and cancer research. Sir had membership in various professional bodies. Now I request our honorable speaker to continue with his guest lecture.
First of all, am I audible to all of you? Am I audible to all of you? Hello.
Sir, very good morning, sir.
Very good morning. Am I audible to crystal clear to all of you?
Yes, sir. Sir, your voice is very bit low, sir.
Hello. Am I audible to all of you?
Yes, sir, but it's bit low, sir.
Okay. YouTube is ongoing. So one thing I want to highlight. So please do not continue the YouTube. Simultaneously, YouTube is going on. So kindly YouTube is going on. Kindly look into it.
Sir, we are not clear, sir.
Now it is okay. Now it is okay.
Yes, sir.
Okay. Sorry for the initial technical glitches. And first of all, I would like to extend my sincere thanks to Vishnu College of Pharmacy for providing this opportunity to talk on these important topics. I think this will benefit to all the fraternities of pharmaceutical sciences who are the budding pharmacists as well as the faculty members of this pristine institute. One more thing, the pharmaceutical research have been gone so far in this new dimension, and there are ample of new molecules are coming in the market, as well as new formulation designs have been carried forward in this dimension. So we have to prioritize, and also we need to emphasize on the new type of research, so our students get excellent excellent exposures to the new kind of research. At the same time, so that they can develop new technologies in upcoming days. So I also once again thank APTI and the Vishnu College of Pharmacy to have a pioneer in the field of education, because they have understand the sense the importance and significance of targeted therapy, and because this is the future indeed, and now new technologies are coming. Like today's I'm talking about the liposomal technology. In that also, depoform technology is one of the key component in the on which many companies are working constantly to develop such a new dimension of technology to elicit a very good quality drug delivery system. Okay, so with your prior permission, I would like to share my screen right now. Shall I? Shall I share my slide? Hello? Hello?
Hello.
Yes, yes. Shall I share my slide now?
Yes, sir.
Okay, thank you. So is my slide is visible to all of you?
Yes, sir.
Okay. So first of all, good morning to everyone. Today we are going to learn about the liposome as targeted drug delivery system for the cancer therapy. There are many research in this dimension have been going on. And the oldest molecule like paclitaxel, docetaxel, many kind of formulation in the this new like BCS class 4 and BCS class 2 drugs who are having a low solubility. These kind of drugs are actually initially have been targeted by the liposomal technologies because it's having a well-versed what can I say, drug delivery as well as it can be encapsulated properly. However, in the chronological order, the development of the liposome have been exponentially increased. Now, liposome is a parent molecule. After that, neosomes, endosomes, phytosomes, bilosomes, puffasomes, then cubosomes, many other substantial lipid-based derivatives have been come across in the market, and new research also going on. Right now, I'm because I already had some research on liposomes. I am also working with the ufazome, and last year also I have published one paper on ufazome technology. These ufazomes are also having a very good impact on the drug delivery system. But since we are restricted to the liposomal technology for today, so first we should know about the targeted audience should be able to understand what do you mean by liposome? What are liposomes basically? So liposomes are colloidal particles formed when a phospholipid is hydrated. So there are multiple lipids we know about phospholipid, then triglycerides, and many other lipid constituents are there. But phospholipids are having a very good priority, prioritized because this actually can assemble in such a manner that actually create a hydrophilic tail, and that the tail will be assembled in the one dimension, and its head will be substantially arranged in the one dimension. So it actually create a spherical out there. So due to the formation of the spherical within the tail within the tail, the pharmaceutical substances as well as chemical moieties can be encapsulated. Chemical moieties can be encapsulated, and further the entrapment efficacy can be improvised. Those who know about the entrapment efficacy, I want to highlight that if the drug is not having a proper entrapment within the formulations, what are the formulations? Liposome is there, one for type of formulations, or you can say the carrier, a drug carrier. So if it is not having a proper entrapment efficacy or entrapment, then the drug will not be properly delivered to the systematic circulation. In the course of time, we will learn about how the drug is being delivered in the system by through the liposomal technology and how the target specificity have been improvised. Now, coming to this liposome, basically, if you can able to understand that basically they are actually colloidal particles, and it is form of as I rightly just telling you, that is actually phospholipid is hydrated in excess of water, resulting in the formation of liposome of size range of 0.1 to 0.5 micrometer in diameter, and they consisting of aqueous pore entrapped by one or more phospholipid bilayer. So as you can see over here, the right side picture, it is comprising of the phospholipid bilayer. Inside of the core, there is another core where you can entrap some other molecules. So in the simultaneously, you can also have an entrapment of two type of drugs, hydrophilic and hydrophobic. Both the things can be done, but in a new technology, which is called as depoform technology. So depoform technology, by which we can have a multivesicular system. In the multivesicular system, so you already know about these things if you have a basic understanding. So multivesicular systems of derivatives have been widely used by the depoform technology. So liposome, due to their basic actually biphasic environment, it can act actually carrier by hydrophilic and hydrophobic, both the drugs. So what you can do, you can see one thing, there are inside there is a specific pore. In the spore, you can have another drug, and outside layer, which is actually phospholipid, you can have another drug. So simultaneously, both hydrophilic and hydrophobic, both of the drugs, you can have interactions, and you can have an entrapment out there. Liposomes gains a lot of interest in advanced drug delivery because of their nature of structure and encapsulation, both of the hydrophilic and lipophilic drugs, as I was rightly telling you. You will be surprised to know one journal called Taylor and Francis, Taylor and Francis journal named Liposome. There are substantial researches going on in this dimension, so they have introduced one specific journal called Liposome. So that shows that there are many researches going on in Hyderabad also. In Hyderabad, Dr. Reddy's company is working hard in the liposomal technology, and they are also giving technology transfer to the other subordinate company as well. So liposomes really indeed carrying a very good significance in the long run of targeted drug therapy. However, I am not going to now the targeted specific. First, you basically understand what is meant by liposome. Then we'll go with the substantially with the liposomal targeting. The biocompatibility as the natural drugs and the targeted moiety, their surface for the stability. So that is the basic things I want to highlight. If you don't know about the targeting, let me explain you. See, our body is having a protein lipid protein trilayer. Now, the protein lipid protein trilayer, that's having a specific significance. Like as you know, the proteins are having a amino derivatives, one amino derivatives, one carboxylic derivatives, one CH3 group, and one OH group. So now, subsequently, because it is a completely outcome of amino acids, because proteins are actually the main building blocks of any protein is amino acids. There are 21 amino acids are there. So now these amino acids can go for a specific carbamide coupling reaction. So that is only possible once you phospholipid. Actually, the outer surface, you can see the round color small molecule, that round color shapes balls. This in that the target specificity can be improvised, where we can graft some kind of ligands out there, and so which these molecules that can go to the phospholipid, that become more target specific. So now, what type of ligands we can add? We can add subsequently your enzymes, or your hormones, your anti-epidermal growth factor, some kind of receptors we can add out there, and then subsequently we can add to this molecule to the systematic circulation. So there are many things. So the basic part of delivery, just listen to me very carefully what I'm going to say. The basic part of the delivery of any liposomes, depending on the essence of the condition, or the condition as well as the physiological state of the particular places. Like I was talking about the delivery. Well, suppose you do have a capillary which is completely perfused with some unwanted subplaces. I'm talking about any kind of cancer. So in the cancer, you can have a multiple proliferations. Okay, multiple proliferation and abundant capillaries you can see. So during this capillary, because once the cancer is developed, there is an unconditional and exponential growth of certain cells. It is not been controlled by any kind of machineries. So it has not been properly controlled. So therefore, there is a development of the cells out there. So once it is developed, the capillaries of the blood capillaries also, exponentially concord the cells. When it is happening, then there is some kind of perforations, or there is some kind of breakage in the capillaries. And due to this thing, these capillaries are fragmented. And from the capillaries, the drug particle can be encapsulated and enter inside of the particular cancer cell, and this is called as that is called as enhanced permeation retention effect. That is called as enhanced permeation and retention effect. So there is a will, there is a way. So scientists have actually come forward, understanding the enhanced permeation and retention effect by through which they are actually targeting the liposome-based targeted drug delivery. So this is the basic core structure of the liposomes. Now we will see the classification of the liposomes. We are talking about there are large liposomes, gigantic vesicular liposomes, then you small unilamellar vesicles, multilamellar vesicles, and multilamellar vesicles, and so many other examples are already there. During this process, after the class, after these things, I will also teaching you how to structure and draw such liposomes in our softwares. That also I'm going to teach you. So stay tuned to this presentation. So now the basic structure is one lipid layer. One lipid layer. We do also have several bilayer or lipid bilayer lipids, which is multilamellar vesicles. And these multilamellar vesicles can also encapsulate small unilamellar vesicles also inside. So that become more productive and proactive, where you can target multiple products out there. So now coming to the basic composition of any kind of liposome, it is actually conventional liposomes. We have a fusogenic liposome, pH sensitive, cationic liposomes, long circulating liposome, and so on. So you can see out there, if you increase the if you can increase the concentration, or if you can increase the diameter, it actually can encapsulate small liposome as well inside. So liposomes are this already the many kind of products actually going on as a nano-targeted drug delivery. You might be knowing about the polymeric lipid hybrid nanoparticle, polymeric nanoparticle, nanoarchins, then mesoporous silica nanoparticles. But none of them actually established and developed in the completely developed in the clinical trials, and therefore they are not being sensitized further. However, liposomal technology is having taking a great share of the market, because this technology is well established, as well as it is clinically approved. And therefore, as I rightly said previously, the Dr. Reddy's is already established a lot of research. They have incepted a lot of research on this liposomal technology development. Now you can see the first picture, the A. You can see the lipid bilayer. You can see the lipid bilayer arrangement. Inside of the lipid bilayer arrangement, you can keep the hydrophobic compound out there. So hydrophobic compounds can be encapsulated. So what are the hydrophobic compounds? As you know, we know the BCS classification, biopharmaceutical classification 2, and biopharmaceutical classification based 4, the taxol, docetaxel, paclitaxel, okay, doxorubicin, so on. Many others derivatives are there which can be encapsulated in the hydrophobic compounds, where it can be stabilized properly, it can be retained further. However, in the inside of the core, we can have the aqueous core, where we can have another drugs. Okay, those drugs which are having a synergistic property, their CT value, the cumulative cumulative synergistic values are, if it is good, almost around almost around 2.5 or so on. So then these synergistic products can be encapsulated inside, or certain drugs which are adjacent to each other. Suppose you are talking about melatonin. Melatonin is a hormone. Melatonin is a water soluble. So you can keep the melatonin inside of the aqueous core of the liposomes, and the surroundings you can have a paclitaxel, docetaxel, which will be in the hydrophobic compound. As a hydrophobic compound, that will be in the lipid bilayer. So simultaneously, you can deliver two drugs in a single go. And at the same time, since it is actually mimic the exosomal environment. What I'm talking about the exosomal environment is body is having some exosomal behavior, like it is actually mimicking the exosomes of inside of the body, the extracellular fluid. So it can easily penetrate by the active transport, facilitate diffusion, and passive diffusion. We will be learning this stuff by pictorial dimension within short, how the flow of the liposomal targeting is going on. So what we learned up to now, there are multiple, gigantic unilamellar vesicles, large unilamellar vesicles, small unilamellar vesicles, and subsequently several layer vesicles are there. And also we learned there are having a multiple dimension, like 5 to 5.5 micrometer, then 2.4 micrometer, and 100 micrometer. And you can, even though we can smaller parts of flow up to around less than 50 nanometer, also we can be carry forward to prepare the liposomes. And at the same time, the liposomes can be targeted for hydrophilic and hydrophobic drugs. So suppose another example I want to imatinib mesylate. Imatinib mesylate is an anti-cancer drugs which can be encapsulated as a BCS class 1 drugs. So BCS class 1 drugs is basically high soluble, high permeable. That can be encapsulated inside of the liposomes and the surroundings, which is having some imatinib having some adjacent, you can say some synergistic activity with capacitabine or some other drugs, which are BCS class 2 drugs. That can be BCS class 2 and BCS class 4 drugs. That can be encapsulated in hydrophobic compound, where the lipid bilayers are there, where you can encapsulate. Therefore, we can be delivered dual targeting possible, hydrophilic and hydrophobic, which is not completely possible in the normal drug delivery as well. So what are the advantages? We have learned about the basic concept of the liposomal technology. What is the advantage? They protect the encapsulated drug from the metabolic digestions. So one specific process called reticuloendothelial opsonization process. Reticuloendothelial opsonization process means in our body, we do have some opsoning glycoproteins. These opsoning glycoproteins are so teeny, so fragmented and so small that actually if it is having a 200 nanometer range, like that can easily crispy that can be target, and it can engulf inside by the exocytosis, and the further pinocytosis can be taking place. So basic composition of the cells, if you can know that inside there will be nucleus, then cytoplasm, then extracellular fluid, and so on. That is the mitochondria, then subsequently extracellular fluid and intercellular fluid. So the basic business of our liposomes is to how to enter from extracellular fluid to intercellular fluid. But when the extracellular fluids in extracellular fluids liposomes reaches, there are possibilities where opsonin glycoproteins can be encapsulated and it can destroy the cells. And there are certain other challenges also which can be overcome, that need to be discussed, which are actually because you know that our body is having a very much blood flow inside. Very fastly the blood is going on. There are substantial possibilities are there. The drugs encapsulated inside of the liposomes can be destroyed, and that can be fragmented further. So the protected encapsulated drug from the metabolic digestions is one of the key components. That's why we need to prepare a product so that it can be encapsulated properly, and therefore that it can be carry both the lipid and water soluble drugs. As I rightly said in the previous slide, that it can be both the lipid as well as the water can be encapsulated. Water soluble drugs can be encapsulated inside, and subsequently can be target the specific tissues and the cells. So specific tissues and the cells, if it is not been targeted, then what is the use of liposomes? Because it is a more target specific. And you will be surprised to know the burning question of now new dimension is the cancer. There are multiple cancer and people are suffering from angiogenesis. The angiogenesis means if the body is having anywhere you do have a malignancy. Suppose you are having a colorectal cancer. That colorectal cancer can be spread in the entire body by the systematic circulations, and that is called as angiogenesis further. So that's why people are really scary. The people are scared about the cancer, and it is abundantly increasing because of our food, digestion system. We do have a very worst. Nowadays we are having melted tension. Once I was doing my postdoc, I come to know that is a melatonin. I was working on the melatonin molecule. I come to know it is acting on the circadian rhythm. But however, this melatonin, if it is fluctuated because of mental tension and because it is helping on the heating cooling cycle of the circadian rhythm, if it has been fluctuated, the people also suffering from some kind of pineal body cancer. Many kind of things are there. So what I want to say is that there's an alteration in the hormonal alterations and the body have been changed, and then subsequently the cancer is a palpable significance, a palpably it will be coming. So therefore, the target specificity of tissues are very much important, and where liposomes are giving these advantages. And at the same time, can make into the variety of size. We can manipulate the size. We can create a multiple size out there. Then reduce the doses. We can reduce the doses because we become more target specific. Target specific means we don't we deliver the drug which is required. We don't want to deliver much more drugs. And most of the time, the Taxol and other derivatives partially potent. They are very potent drugs. They are really potent because, as you can see, that since if it is not been target specific, what are the complex phenomena you observe? You can see the people who are not giving a target specific, they're just giving an injection of any molecule. They do suffer from secondary alopecia. Alopecia means a baldness of a hair, because maximum cancer patients, you can see the baldness, not only baldness, their hair follicles as well as their nails become fragmented. Why it has been happening? Because the cancer and anti-cancer molecules are targeting all the proliferative cells. They don't differentiate what is cancerous molecule, what is other cancerous cells, and the proliferative cells, normal proliferative cells. They target everywhere. So that's why it is very much important to have the target specificity, so that you can reduce the chances of alopecia. You can reduce some myelosuppression. Myelosuppression is another one. The bone marrow depletions can be overcome by the target specificity. And therefore, one is the best example is the liposomal technology, which actually reduces the dose, as you know, the reduced dose and more target specificity. The biodegradable and biocompatible and non-immunogenic, they become biocompatible and bioimmunogenic, and possibly several route of administrations can be possible. Act as a scaffold of additional agents. So scaffolding means surface anchoring. The surface anchoring is called as scaffolding. The surface anchoring means you can anchor any kind of protein, ligands, and subsequently other molecule as well, where you can actually target the molecule out there. As I already told you, you can have some kind of proteins, you can have the hormones, you can have a anti-AGFR or tyrosine kinase alpha inhibitors, then subsequently kinase inhibitors. You can encapsulate it out there. Why I'm talking about these things? These things can be regulate the down regulation and up regulation of the cancerous pathways. And therefore, the target specificity much more improvised. It will be working on the cell surface. It will be working on the cell surface. And now, subsequently, the cancer molecule, which will be encapsulated inside of the liposomes, can be encapsulated inside, and it will go to the derivatives. One of the company called Cipla is working extensively in the liposomal technology, and we are blessed to have some kind of material today to show you all how they work in the long run of liposomal technology. So we can have that. So this is very common to be found in amphiphilic nature. They become more amphiphilic, like lipophilic and hydrophilic, both amphiphilic in nature, the liposomes. So we can have a wide range of variety of drug delivery we can postulate in the liposomal technology. So reducing the systematic toxicity, like Taxol, as I rightly said, Taxol, amphotericin, we can do. The increase the efficacy and their therapeutic index can be improvised. Obviously, the therapeutic, if it is retained and sustained in your solid, blood plasma concentration, if it is increased, then subsequently the therapeutic index, the AUC, also can be improvised. The indexing from minimum effective concentration to maximum safe concentration, that is index also can be improvised. So obviously that actually completely help us in the improvement of the pharmacokinetic efficacy, and it's also helped to reducing the exposure of the sensitive tissue the toxic drugs. So these are all the many various examples on which we can target. So up to now, we learn about the different type of liposomes. We also learn how to encapsulate. We learn what are the things can be encapsulated inside and surface scaffold. What are the advantages of liposomes? Now we'll be learning about the preparative methods of the liposomes. So there are multiple methods which are been widely utilized in the liposomal technology. One of such kind as hydration phospholipids and solvent cephalose methods, then hydration of proliposomes method, supercritical fluid method, and freeze-dried double emulsion methods are widely utilized in this dimension. Now we can see subsequently the hydration method of phospholipids. So hydration method of the phospholipids, the method includes the depending of the phospholipid onto the substance from a soluble and organic solvent such as chloroform or ethanol. So liposomes, I'm 100% sure about in the round bottom flask method or handshaking methods. BFAM and FAM students have already worked on this dimension. They have already seen some kind of work in the preliminary. At least they should know about. They are actually well known about how to prepare the liposome, and they do also watch and perform the experimental work like entrapment efficacy, and also they perform the vesicular size diameter determination, and also they can go for a HPLC method to estimate the the encapsulated products. So I'm sure about you have knowing about this, but again I'm reiterating the same thing because these methods are somewhat challenging when you go for industry, because I was talking about the depoform technology, which is widely accepted by the industry, where multivesicular systems they will be utilizing. One of the things what I want to highlight in the industry, what they are doing right now, microbubble technology. Microbubble technology, by the microbubble technology or nanobubble technology, they are developing liposomes. Because obviously these liposomes are vesicular size, and microbubbles technology liposomes are much more, which is not not included in any text because it will be always in the research. Though these things are very much microbubble technologies are basically utilized in the heart diseases and so on. But nowadays, it is also scientists are working on to develop liposomes in this dimension as well. So for the preparation of the liposomes, what basic ingredients we know? We need to have a cholesterol. We need to have certain amount of the organic solvent like chloroform, methanol, and so on. We also need to have phosphatidylcholine, that is, you soil acetate. We must be knowing about these things and what are the required things. And also, sometimes if you go for a hydration method, we need to have a potassium hydroxide and sodium hydroxide and so on, which can be actually hydrated the substances. So the methods including the deposing the phospholipid onto the substance, solution, or organic solvent we need. Then subsequently we need also to form a film. So films, we already know the film formation, the outer layer of the liposomes are prepared by the phospholipids. So bilayers that can be hydrated, absence of the hydrodynamic flow. As I told you, the hydrations can be done by the potassium hydroxide solution and sodium hydroxide. And for days obtained through the aqueous suspension and giant unilaminar vesicles can be obtained. So one more thing, basic components if you want to is to prepare in a liposomal technology in your home or in your labs, how you can do this. Basically, you can consider some phospholipids, then organic solvent, then subsequently mix it well. You can go for if you don't have, then you can for a handshaking method, or else you can go with that is you can go with this round bottom flask methods. In the round bottom flask methods, you can dry the layer. Then subsequently, after drying the layer, you can go for hydration with the potassium hydroxide, and then go for a probe sonication so that the laminar vesicles forms a small one. Initially, the formation will be in a layer form. The phospholipid will be the layer form. Unless and until you go for a probe sonicate, that layer will be started to sandwiching or crisping itself to form a spericular, or you can say the spherical shaped products. So unless and until you don't go for probe sonication, you won't get the product. Only the hydrated products you'll get, but you need to go for a probe sonication or homogenization techniques further, so to get the developed things. So there are things like even though the multilaminar vesicles make the large portion of the vesicle portion, hydrated method also known to the swelling method. So we need to know about the hydration also. This is very, very important. So what I told you, initially you need to take the certain amount of cholesterol. We need to have a swell acetate, and we need to have a certain amount of, you can take any other substances like stearic acid or any other lipids we need to take. Then subsequently, you need to add some kind of what can I say? Suppose surfactant like phosphatidylcholine, triacetine. Another molecule is called as triacetine. Triacetine we can take, and subsequently we can go for handshaking method, or we can go for your round bottle flask methods, where you can go for a drying of the stuff. And once the film formation develop in the round bottom layer, then you can go for a hydration. Hydration with the sodium hydroxide and potassium hydroxide, whatever the availability and based on the requirement. You go for hydrated the substances. Once again, shake it well. Then once the solvent system develop, or semi-transparent solvents have been developed, then go for this one, the probe sonation or sonication. You can go for probe, or you can go for bar sonication based on the availability. Once we go for the sonication, automatically the formation of the liposomes will be taking place, because initially the layer is developed like this. Once you go for a sonication, the layer formation will be in the spherical shape. It will be in a spherical shape, and the drug is getting encapsulated inside. Okay, so there is also always possibilities. Whenever you go for a drug encapsulation, you should be remember that organic solvent consisting of your organic solvent must have your drug which are hydrophobic in nature. So that things you can keep the organic solvent. Make sure organic solvent should dissolve the certain substances. And hydrophilic, the drug in that you can, for the hydration method, you are using some potassium hydroxide water, right? So in that you can add the your drug which is hydrophilic in nature, if you wish to encapsulate two drugs, because you know that as an ingredient of liposomes are basically amphophilic in nature. That means we can have possibilities to conjugate both of the drugs. So that's the main advantages of liposomes, and that's why people are working together to develop such products. Now this figure has been developed by Hello? 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 Shall I continue?
You continue.
Okay. So solvents, spanules methods, that is actually, these figures have been developed by me. So that actually, I will also teach you how to develop such kind of figures. Nothing to panic about. There are some softwares where on which you can develop some figures. So in that figures, you can see that certain conjugation, the passive targeting polyethylene glycol, and subsequently some conjugated products, and then surface conjugated drugs, and also DNase, siRNAs, the lipoplaxis derivatives, and then hydrophobic drugs and variant vectors can be encapsulated. So inside. So these are all the various targeted side on which you can have this kind of things. So in the inside of the liposomes, we already know the small laminar vesicles. We do have a large unilaminar vesicles, giant similar unilaminar vesicles, and multilaminar vesicles, and subsequently we can elevate the system. So these are all the potential places on which the target possibilities have been there. So the solvent spanules methods and methods produce an oil in water emulsion spanules of the lipid-based solvent after rigorous making of the organic solvent. So we need to have this organic solvent. We need to select the specific and precise organic solvent, and evaporation is the used to extract the organic phase and resulting in the covalent and conversion of the spanules in the multilaminar vesicles. So there are multiple things, and that as well as rightly saying about that, this is the basic studies of hydration pro liposomes preparation. So initially we need to create a lipid. As you know already, I said about the lipids. What are the lipids you can have? So that lipids and then subsequently carriers. Then you can have a phospholipid pro-lipid liposome formations with round bottom flask methods. Then subsequently we need to hydrate it. Make sure the hydration will be taking place with potassium hydroxide. In that, you can add the water along with this one, along with your aqueous solvent, aqueous drug, and subsequently you do have go for a swelling. After the swelling have been done, because the phospholipid is started to exponentiate itself, exponentially increase themselves, and subsequently go for agitation. So for agitation purpose, what I said about to go for probe sonication or I have, or you can say fast sonation, you can go with. But I'm not interested to go for the homogenization. You know why? Because maximum, if you go for a homogenization, the particles are getting fragmented. At the same time, the rupture will be taking place. The complete work will be gone vain. So that's why you can suggest to go for if you want to have a large unilamellar vesicles. Then you can go for a batch sonicator. If you want to have a small unilamellar vesicles based on your requirements, go for a probe sonication. So probe sonication also, you should not go extend up to around 5 minutes, 15 cycles, 5 minutes. Don't go more than that. If you go for it, then obviously due to the heat generation due to the probe sonication, the drug will be cooled out, and subsequently your work will be go in vain. So that's why this is the thing you should do it. And subsequently, when you go for agitation, normal agitation from sonication, you do, then you will get a proper outlet. And also you can go with the extraction method as well. Then small unilamellar vesicles is very, very important. That's why, as I rightly said, sonication is important. So once you go for a specific agitation with the normal handshaking method and all, the layers, the hydrated one, which started to arranging itself, and then slowly after the sonications, they started to getting in a structural form. It started to get itself a structural form. So this many kind of research in this dimension is going on to improvise the technology. One of such kind is called as supercritical fluid methods. In that case, supercritical fluid combined with the best quality of liquid and gases, on which you can have the carbon dioxide is the most commonly supercritical fluid, which has been utilized in the study. And this composition mixed with the phospholipids and supercritical fluid and organic solvent is injected to the aqueous process through the nozzle. So as I told you previously, that microbubble technologies is a one part of descendant of the supercritical fluid technology. So in that case also, you can get a very precise and accurate kind of formulations on which you can get a actual, actual size of the formulations. So there are multiple work is going on in this dimension. And I'm sure in upcoming days, we can have a more qualitative work in the supercritical fluids development. You can see that. So this one is partially related to fluid at break process. If you could remember the fluid at break process, the fluid, I'm not saying that this is actually the same, but mechanism is partially same. So in the fluid at break process also, you can get the some kind of flow from the down. Here it is not from the down, but up. But it is a specific gas, the carbon dioxide, where the agitation will be taking place. But make sure the agitation should not be very much sharper, so that the particle should not be get break. And then subsequently, once you added the carbon dioxide there, then it can be started to encapsulate the product, and then by those specific nozzle and the filter. After first of all, you need to go for a filtration, and subsequently nozzle. By the nozzle, you can have it as based on crystal clear, as small unilamellar vesicles, you can able to formulate by the supercritical fluid technologies. So now next step is called freeze drying technology. So in the freeze drying technology, you can have multiple work because many of us might be knowing about the cryopreservation technology. To know more about the cryopreservation technology, you can see some of the book chapters and some review articles I have written of the cryopreservation technology. So this technology is talking about how to preserve and store biological products. Because you know what, whenever you started to go for a lyophilization, what happens? Crystal formation take place. This crystal, once you develop the crystal within the product, you can know the sublimation technique is one of the kind. In the sublimation technique, you know that crystal formation is developed. So this the elevated nature of the drying, freeze drying is the sublimation basically. So sublimation technologies, when the sublimation is taking place, then subsequently the formation of some crystal is developed. These kind of crystals are actually lethal to the particular product. They actually rupture the phospholipid layers out there. And if it is ruptured, then what will happen? Then obviously the drug will be pulled out from the inside of the encapsulated product, and then there will be no use. You will be see the separation of the product. There will be a phospholipid separately, the supernatant separately. So that is we don't want. So for that reason, the freeze drying technologies were being utilized. This freeze drying technology, how you can use in the presence of suitable cryoprotectant or cryopreservation technology. So in the how we can do for a cryopreservation, what is the use of cryopreservation? The cryopreservation is talking about the mannitol, tetrahalose, sorbitol, this kind of, even sometimes one patent is there for sodium chloride. This actually retain the vapor pressure of the molecule so that it will be not uh, it will not create any surface crystal once you go for a lyophilization. Many of us know about the lyophilization or freeze drying techniques, one which the minus 85 degrees centigrade, the things will be grow. And once it is been dried, then what happens? The crystal formation develop, and then it will be destroy the cells. So to prevent this crystal formation, we can have a specific cryoprotectant or cryopreservatives. Once you add, then integrity of the solvent system and the integrity of the product will remain as such. And the biological product, if you can have the, as I told you, there are hormones, there are anti-GFR or tyrosine kinase alpha, which can be encapsulated or surface scaffold over the surface. So that will, their integrity remain intact. So therefore, the cryopreservative freeze drying is one of the great technology to emulsify and produce your liposomal technology. So in that case, freeze drying liposome forms on the water soluble carrier and dissolve tetrabutyl alcohol. We can have the co-solvent system, and then when the water added to the freeze dry, it produces the multi-amino acids. And in that case also, when you prepare the product, you can have a double emulsification method. Whenever I said about the double emulsification method, that is actually water in oil in water technology. So you can have a multiple process so that it become more stable and more fragile, not fragile, but become more stable. The stability is more important. So since we are progressing towards from the small unilamellar or simple method to the larger technology, so that's why it is very much important to have the multilamellar and you can have the double emulsification methods, and subsequently liposome produces smaller than 200 nanometer and encapsulated efficacy varies depending on the species encapsulated. So these are all the yes. So now let's come to the actual picture. So you can see the liposomes in the cancer therapy. If you want to have the cancer therapy, what are the things we can have? We can have a targeted ligand, functionalization of the substances. We can have a pegylation with the polyethylene glycol. We do have a small molecules. We can have a positive charge cationic substances. We can have a negative charge anionic substances. Then subsequently, hydrophilic drug molecules can be encapsulated, and hydrophobic drug can be encapsulated in the phospholipid, the tails, within the tails, we can go have a some kind of substances. And once again, the pegylation. If you go for a pegylation of substances, I don't want to go in much more detail, but I want to tell you the stealth liposomes developments can be developed. So stealth means they cannot able to identify, but the bloodstream will not be able to identify them. They cannot able to rupture. They cannot able to rupture the substances. They cannot able to rupture the substances. So that's why the liposomal carrier systems are being widely utilized. And then there are certain things like phospholipids, cholesterol, and hydrophilic polymers can be utilized, and hydrophobic polymers and polyethylene that, as I told you, the pegylated liposomes can be utilized. The phospholipid complex in that, just a moment. So phospholipid lipids are anti-cancer drug derivatives and intracellular with the liposomes with the increased surveillance time of the encapsulated that is polyloaded and cancer is being encapsulated. The cancer products can be encapsulated inside. As I told you, the cholesterol is one of the key component, and hydrophobic payloads need to be given in the inside. So this is give you a complete picture of how we can encapsulate and how we can do this. And these are all things that are possible in our lab scale. There, if you at least the core structure we can develop, and subsequently, if you want to add a layer out there, we can have a more core out there. We can keep a certain other substances inside. So hydrophobic payload and polyethylene glycol loaded lipids can be encapsulated to prepare a stealth liposomes. Stealth liposomes are much more much stable, and the reticular endothelial opsonization process and other substances will not be affected accordingly. Now we'll talk about the active targeting. The targeting of the liposomes, which we are now going towards the targeting aspect. As you can see, the outer layer of the any kind of cells having developed some kind of over projections. This projection is called as epitopes. These epitopes are getting targeted successfully with the various site by body accomplishing the various form of the using the drug carriers. Different ligands may be functionalized in scaffold on the surface of the nanoparticles and allow the antigens expressed on the cancer surface to be targeted. By linking with the disruption and the targeting reticular endothelial process, immunoliposomes may be actively targeted. So we can have an immunoliposomes. When I was talking about the immunoliposomes, that means it can be encapsulated and surrounded by certain kind of lipids, as well as so obviously, it can have certain anti-EGFR, monoclonal antibody, and some other substances. Polyclonal antibody, lipoplaxis, polyplaxis can be these kind of things in my terminology you might be knowing about. Then aptamers can be also been targeted accordingly. So there are many researches going on, as I already told you, that EGFR, that is, human growth factor, and subsequently PK13K, AK2, then MP8K, some kind of receptors targetings also can be possible by through this process. Then antibody-mediated targetings, that this is the one which maximum companies are working. The antibody-mediated targeting is a common form of the targeting, since the antibody fragments have a better affinity and more excellent selectivity for the cancer cells. When these antibodies-mediated liposomes interact with the cancer cells, they are engulfed. The engulf means it has been completely captured and it will destroy. Engulfed, okay, inside of the cancer cells, and process called as endocytosis. So we know that by endocytosis, the process on which they can be encapsulated, which simultaneously causes the epitopes, so apoptosis. So this the apoptosis is nothing but the destruction of the cancerous cell. Okay, it can be chronologically, it can destroy and kill the cancerous cell without harming the normal body cells. So this is a very, very important term, without harming the normal cells. What we learned that if we develop a specific mechanism on which you can prepare the liposomal technology, so that liposomal technologies can be these liposomal technologies can be not to target any kind of cells, but to target the cancerous cells, on target to the any specific cells. It will not destroy that. Therefore, the chance of secondary alopecia will be minimized. So antibody plays a vital role, allowing the specific recognition of the cell surface protein and receptor cells and member of the cells. So another approach is called as targeting. This diagram has been developed by me, so I want to highlight very specifically how it is look like. So there are different kind of targeted cells. So you might be knowing about there are kind of endothelial growth factor. Even certain that is called as that is adenovector or adenovirus. This is not a specific virus. This is actually a virus shell, adenovirus. You can search in internet, you can find this adenovirus. This adenovirus is basically cell on which you can encapsulate the targets out there, and then there will be some outer layer, or by through passive targeting, within the cell, outer surface of the cells, the diagram, the cells can be encapsulated. Means you can target two adenovirus where the drug can be encapsulated, or liposomes can be encapsulated, or you can have the surface scaffold of the liposomes by adenovirus, and it can be encapsulated, and it can be delivered in the cell inside of the normal bloodstreams. It can be release the bloodstream. So when the particle size of the liposome nanoparticle which matches 85.5 nanometer, then this kind of activity has been possible. So longer run, not the multilamellar vesicles or large gigantic lamellar vesicles are important, but the small unilamellar vesicles are important. You know why? Because reticular endothelial opsonization process, which actually targeting the 200 nanometer range particles above. So we can have particles which are below 200 nanometer particles, so they are can able to go through a reticular endothelial. They can pass through the reticular endothelial opsonization process. At the same time, that will be participating in the cancer targeting. Moreover, if you can prepare a still liposome-based product with the pegylation techniques, that also create a very much significant impact of the liposomal targeting. Another approach is, as I told you, by the adenovirus technology. In the adenovirus technology, it has not been developed in India. In India, Hamdard, Jamia Hamdard is trying to do so this work. Jamia Hamdard, Delhi, but they have failed. I personally know some people they failed it. But adenovirus technology means they want to collect the virus shell, and inside of the virus cell they can encapsulate target specific liposomes inside, and then they can deliver by passive targeting. So this is one of the projectile of how it will be taking place. Now, aptamer-based mediated targeting. That aptamer-based mediated targeting, as I already told you, that is fundamentals. This aptamers were the first introduced in 1990, where the single-strand nucleosides are considered as the novel class of ligands widely. And these aptamers can be conjugated with the siRNA, and then subsequently, moreover, they have a minimum side effect. So this siRNA and protein-based targeting on the outer surface, that can be a wide range of work. When I was working on IT, so I have seen that many of us, of our colleagues are working on this dimension, and the protein aptamers-based conjugation are and siRNA conjugation, RNA conjugations are one of the significant prioritized in this dimension, and many people are working on this aspect. But still, in India is infancy. Because of after the preparation that need to be also encapsulate and so on, it can be freeze dried. So now next is your protein and peptide mediated targeting. You can see that pegylations, siRNA, peptide excels can be encapsulated and then it can be stimulated. So since we do have a constraint of time, I'll be going very fast because I think I'm awarded only 1 hour. So I'll go a little fast. But that's the thing I want to highlight. I want to ignite inside of yourself that you should be working on the target-based drug delivery, where you can target a lot of things like siRNA, polyethylene glycol conjugated products. Also, you can do. You can have a adenovirus encoded, as I already told you. The adenovirus targeting them, you could do have a better proteins are also there, doxorubicin, and many of the work you can do accordingly. Now we'll see a video which actually talking about how the encapsulation has been taking place and how the formation was taking place. And thanks to this video, thanks to Sipla. Okay, so I have almost I have covered all the content, but since we do have a constraint of time, I would like to extend that the future perspectives of the liposomal technologies is bright and shiny. So that's why the mechanism by which liposomes will be penetrated to the tumor site need to also work on it in future. And with the advanced technology, we believe, and I personally believe that is possible. So there are many research which is going on in the literature of the patents. Many you can see out there. I'm not going into details. Many research has been going on and patents have been going on. Patents have been filed in this dimension where the liposomes technology is widely used, and many other research is in ongoing in this field. So these are all the various kind of research activity as well as the liposomal technology was being utilized. But in the college, I must want to tell you, if you wish to go for agitation of any preparing of the liposome technology, do not go for a high shear homogenization technique. Go for a high pressure homogenization technique. High shear means any kind of homogenized, you can say tissue homogenizer and any kind of stuff. Don't go with that. You can go for a high pressure homogenizer, which is coming with a different, like oxygen gas need to be followed and so on. There's a different technology. And subsequently, you can go for a probe sonegation. So probe sonegation is very much important in this dimension. Okay, due to the time constraint, I will not able to cover these portions. I hope I'll be going to share this slide to all of you. So once again, thank you all of you to listening patiently, and I hope within a 1 hour 15 minutes time, I have ignited the knowledge of liposomes and the target specificity of the liposomes. I personally believe you are ignited now, and you can, as a M.Pharm student, B.Pharm student, as well as the faculty members can work on the liposomal technology as well. So this is our studio, our university, NIMS, Nasimuddin Institute of Management Study, where we are constantly working to develop new set of things. And we do have more than 150 patents with multiple aspect of research. So, and a number of publications are also keep on increasing in this our institute. So thank you one and all. And since due to the time constraint, I could not able to show you one of the things. So just want to brush up. There is a software on which you can work and develop these things that is called Adobe Illustrator. So due to the time constraint, I could not able to take this hands-on training to you. So this is very simple, and also I think you need to purchase this software. This year's software has been given by from our university, that is called Adobe Illustrator, on which you can develop multiple multiple liposomes, phytosomes, endosomes, and so on. So this is the software you can see out there. The same things have been encapsulated and kept incorporated inside of my this thing. That software name is Adobe Illustrator. So with that note, I would like to end up my presentation, and thank you one and all for this opportunity. I am ready to answer your valuable questions if you have any. Thank you one and all.
Thank you, sir, for enlightening us on preparative methods of liposomes and different types of targeting system and their mechanism. Now the section is open for questions.
Hello, good morning,
Yes,
sir.
please. Yes, please.
Myself is Vijay from National College of Pharmacy. My question: are there any techniques that are used to see patients hybrid
You are not. I'm not able to catch your question properly because of technical glitches.
Are there any techniques that are used to see the phase separation between polymer and hybrid?
I'm unable to understand what you are saying. You are talking about the polymer techniques.
Are there any techniques that are used to see the phase separations in hybrid?
Okay. Are there any technique? You mean to say are there any technique to? Okay. There are tremendous background noise. Phase separation. You are talking about the phase separation technique, right? How to minimize, right? See, I want to tell you, sorry for the background noise. I think there is some issue with the background, but I want to tell you that you can have some channelizer out there. Once you want to, there is obviously the possibilities are there that the phase separation has been taking place. So you can use you can have the sodium chloride solution as a channelizer out there. So these channels actually make the product, and also certain extent you can have potassium chloride, sodium chloride, these kind of things, which is actually acting as a channel forming agents on which it will acting on the phases, two phases, once you added this thing. So due to this thing, this process, the formation of the product will be well connected, and there won't be any kind of agglomeration. There won't be any kind of agglomeration. But however, you need to select. You cannot extend the concentration more than required amount. Like at least the concentration has to be not more than 0.5% of the total of the total total solvent system. So 0.5% you can take, and in between it will be acting as a channelizer between the two phases, and then automatically stability has been improvised. Okay, next.
Good morning, sir. Myself, I'm B. Pravalika from Quality Assurance Department. Sir, my question is, if my study of interest is to suppress the tumor size, I wanted to make a plant-based, like vinca type of alkaloid into a liposomal form. So what I do is, to make it as a formulation, I have seen all the techniques you have explained, sir. The most suitable technique I found is hydration of proliposomes. So is this technique useful, or any other technique like freeze drying technique for
I personally want to say don't go with the liposomal technology. The liposome derivatives have been far more advanced right now. You can go for phytosomal or cubosome. You can go for a cubosome techniques. It is actually a descendant of the liposome technology only, cubosome. And also at the same time, you can go with the nanoithosomes technology, nanoithosome technology and cubosomes. These two and phytosomes. 3 things you can target. Okay, that I can say. And one more thing, vinca alkaloids. When you go for extraction, how do you go for extraction of the vinca alkaloids?
Sir, by acid separation method.
Okay, so that actually need to be checked whether the derivatives have been forming crystals or not. If it is formed, once again, I was talking about if it is forming a crystal, then it will be rupturing your product. And you should go with to check whether it has been encapsulated properly. There is a specific technique usually utilized. I was not talking about the characterization here because of time constraint. There are one technique by which Tritonex, you might be knowing about. Tritonex is a solution which will be utilized to lysis the liposomes. So you can go and check the entrapment efficacy by through Tritonex in this dimension. And one more thing, crystality. If it is from a crystal, then obviously you should stop there. So how to improvise not to have a crystality? You can go for solubilizing a proper way of the product. Dental alcohol. You can work dimethyl formamide or dimethyl sulfoxide. These are all the 2 solubilizing agents. Very small amount, small pinch like 0.5 milliliter or something like that. You can add with your vincastin and vinristin combination. Okay, mix it well. Then you can prepare a slurry. Okay, so based on that, you can go for the process. Initially, you have to fabricate with the you have to fabricate, or you can have a mix up with the properly with this dimethyl formamide or dimethyl sulfoxide initially. Why? Because that will create in the long run, like if it is from a crystal, the liposomes won't be developed well. Because if you wish to develop the target specificity, the normal liposomes you are going to prepare, or liposomes or cytosomes or cubosomes or whatever you want to prepare, it will be in the initial phase. Now you want to make a target specificity, you can go for targeting with any type of grafting that is called as normal grafting technology, where you can target any protein, siRNA. In school level or in college level, you can have siRNA, aptamers, targeting, it is possible. However, this anti-AGFR monoclonal antibodies and all, this is a little bit tricky aspects. So that is not possible. So accordingly, you can plan. I want to correlate. Suppose you do have a problem with the solubility of any API. What do you do? Initially, you have to coat with the surfactant, sodium lauryl sulfate, for then so on. So that it's having a proper dissolution profiling and solubility analysis. Same thing, same way. In that case, you first of all separate the your vinca extract, like vingastin, vincristine, then add few drops of dimethyl formamide or dimethyl sulfoxide. You add few drops, then mix it well. Then you can proceed the same procedure. You won't face any challenges on that.
Thank you, sir. So informative.
Thank you. Any other question?
No, sir. As a token of gratitude from Sri Vishnu College of Pharmacy, kindly accept the certificate of appreciation.
Thank you all. I would like to thank everyone for this opportunity. Okay, shall I leave?
You can. Thank you.
Thank you. Thank you.
Closing remarks of Day-3 by Dr. Ashok Tulluri, Organizing Secretary.
Yeah, it is my pleasure to all the gathering and a warm thanks to Dr. Shankar Bhattacharya for enlightening us on the topic of the liposomal targeted drug delivery system, the treatment of the cancer. And the topic is very innovative, and even he incorporated all his research works in that one. And the session is very lively. And what I requested is, if possible, can we share your PPT with our participants? With your permission, we will.
Definitely, sir. I'll provide my PPT. I'll mail you the PPT.
Yeah, then it will be more informative to all the participants, because most of the online participants, they are giving the SMS for us. They are typing the message in the text box. They want this presentation. If provided, please provide the PPT. So they will be more happier.
I'll definitely do, sir. Definitely.
Yeah.
Anything good for pharmacy education.
Yeah.
No problem, sir.
Yeah, thank you very much, sir. And this association, we requested you to continue for our next further program, future programs also. Thank you very
Yes,
much.
sir. Thank you, sir. Thank you.
Thank you, sir. Next, validity.
Valedictory vote of thanks by Dr. Ashok Tulluri, Organizing Secretary.
Dear participants from different states and union territories, on the behalf of our honorable principal sir and the convener, Dr. K.S. Nataraj sir, I am presenting this vote of thanks. Firstly, I would like to thank our honorable guest speakers, Dr. M.S. Muthu, Associate Professor from Department of Pharmaceutical Engineering and Technology, Banaras Hindu University, Varanasi, Dr. Subramaniam Nateshan, Professor, Department of Pharmaceutics from Naipur, Kolkata, as well as our today's guest speaker, Dr. Shankar Bhattacharya, Associate Professor, Department of Pharmaceutics from NMIS School of Pharmacy, Shirpur campus. We are very much enlightened to have you, association with you, sir, and a very nice presentations we received from you. The same is the response from all the participants, around 1,500 participants from 23 different states and 2 union territories of India. And also, I would like to convey the best regards to all the participants from our chief patron, Sri K. Vishnu Raju Garu, chairman of Sri Vishnu Educational Society, and the patrons, Sri Ravichandran Rajagopal sir, vice chairman of Sri Vishnu Educational Society, Sri K. Aditya Vishnu, secretary of Sri Vishnu Educational Society, as well as Dr. Kumar V.S. Nemani, director of Sri Vishnu College of Pharmacy. And also, best regards from Dr. G. Deval Rao, president of Association of Pharmacy Teachers of India, Andhra Pradesh branch, as well as from the HODs of the departments, pharmaceutics, as well as the Department of Pharmaceutical Quality Assurance, who have organized this event. And also, I would like to congratulate all the teaching and non-teaching staff, as well as the student volunteers who have given their helping hands to make this event a grand success. Hope we are having a series of webinars which are coming within a short duration of the time. So everything will be communicated to all the registered candidates through their emails or through the WhatsApp groups. And one more request, every day the feedback form, attendance feedback, as well as the evaluation form, what are being sent to your email, registered email IDs, as well as in the WhatsApp groups. I request all the participants to fill all the 3 days feedback forms. Then only you are being awarded with your participation certificate. Thank you all for your warm support. Keep stay connected and keep learning. Once again, the Vishnu College of Pharmacy is welcoming you all for the future events, as well as thank you for your participation to the current event and making it a grand success. Thank you all. ఎవరు?